أهلا بيكم في دروس الأحياء للصف الثاني ثانوي لغات. Today's lecture we will start a discussion about the respiration in living organisms. So let's begin. About the concept of respiration and its importance to the living organisms. We studied how the organism gets its nutrition, absorbs it, and stores it in complex forms, then to as stores of energy compounds, as high energy compounds. Then when trying to utilize them, he breaks them into simpler forms. Respiration is a process occurs by taking in oxygen, either directly by unicellular organisms or by using a special type of system for aeration, which is the respiratory system, in case of multicellular organisms. And as a result of this respiration, carbon dioxide is produced as a final product of respirations. Now, I don't want you to get this confused between it and gas exchange, because here in cell respiration, the cell breaks down food molecules in order to release the energy it needs, okay? In the other case, it's a simple gas diffusion. Okay. About the cellular respiration, glucose and other carbohydrates are considered as a form of stored energy. This is how the living organism extracts its energy needed for its vital functions. Energy is extracted from chemical in the food molecules, and we will be discussing the metabolism or the breakdown of glucose molecule, because considered an excellent example to study the steps of breaking down of the food molecules, and it's usually used commonly by majority of living organisms. So that's why we will be talking about glucose from now on. And at the end, you can calculate how many ATPs resulted from metabolism of one molecule of glucose, which is 38 ATPs. Energy required by the cell needs ATP, but before that, let's study the structure of ATP to understand how it stores energy. The molecule is built up of three subunits, which are adenine. This one is the adenine subunit. This one is a pentose sugar called ribose, and these are three phosphates bonded. See the bonds between the phosphate. These are the bonds. When broken down, there will be release of energy, and it's usually one molecule of ATP, one amine phosphate from each ATP is broken down to form what's so-called the adenosine diphosphate, or ADP, okay? This is adenosine triphosphate, 3-phosphate. When we break down a bond of a phosphate, so the result will be adenine diphosphate. Okay, this is the structure of ATP and how it stores energy. As I told you before, only one phosphate is broken down, so the result will be adenine diphosphate. Energy from 7 to 12 kilocalories is released, and you can summarize the process of respiration of one molecule of glucose, in this equation. This is starch plus O2. In cellular respiration, the end product is carbon dioxide and water and 38 ATP. Okay, now what happens then? How do you utilize this molecule? In three major stages: glycolysis, Krebs cycle, and electron transport. Now, glycolysis takes place in the cytosol of the cell, while inside the mitochondria, where there are respiratory enzymes, water and phosphate groups, and coenzymes, electron carrier molecules called cytochromes, all there inside the mitochondria for the Krebs cycle and electron transport to take place. So please remember, glycolysis in the cytosol, Krebs cycle, and electron transport inside the mitochondria. During the process of breaking down of glucose and the Krebs cycle, hydrogen atoms are removed. And how are they stored? Taken up by compounds called NAD and FAD. See that? NAD and FAD, they get reduced to NADH and FADH2. These are the hydrogen and the high energy bond carriers that we'll be talking about inside the mitochondria. First of all, glycolysis. It takes place in aerobic and anaerobic respiration. One molecule of glucose breaks down into two molecules of pyruvic acid. As you can see here, this is one molecule of glucose 6C, two molecules of pyruvic acid 2 times 3C. The pyruvic acid is 3C. Okay, this is glycolysis. Passing through glucose 6-phosphate, and here it uses up some energy, breaking down ATP. Fructose 6-phosphate, ATP to ADP as well. Fructose 1,6-diphosphate and 2-phosphoglyceraldehyde. Now the 3C component starts. This is where the structure is broken down into two 3C molecules, okay? And this will change into pyruvic acid after giving off or reducing two NAD molecules and producing 4 ATPs. So the net result will be 2 ATP in the glycolysis. Okay. All these reactions occur in the absence of oxygen. Therefore, these reactions are called the anaerobic respiration. This is how glycolysis forms. The energy resulted is not enough to perform all vital activities, and in the presence of oxygen, pyruvic acid pass into the mitochondria to produce more energy. This is where the Krebs cycle and electron transport take place. Now, the first who studies the Krebs cycle is Sir Hans Krebs in 1937. He makes it into two stages. Each molecule is pyruvic acid. Now we ended up in glycolysis to pyruvic acid. Now this pyruvic acid is oxidized in the presence of coenzyme A into acetyl-CoA. See? Acetyl-CoA. Now two molecules of carbon dioxide are formed, and two NADH are formed as well. Okay. Also, to acetyl-CoA, acetyl groups from the breaking down of fat, breaking down of protein, amino acids, all enter the acetyl-CoA stage to enter the Krebs cycle. Now, each molecule of acetyl-CoA in Krebs cycle splits, the CoA splits off. See? The CoA splits off. And the remaining acetyl group combines with oxaloacetic acid, which is a 4C molecule, to form the 6C molecule citric acid. That's why it's called the citric acid cycle. Okay? The citric acid passes through ketoglutaric acid, succinate, and malate, and again to oxaloacetate to, you know, combine with the acetyl group of acetyl-CoA to form citric acid again, and so forth. 2 molecules of CO2 are released, C1 and 2, and 1 ATP molecule, this ATP, with 3 NADH and 1 FADH2 in each cycle. So in each cycle, 2 molecules of carbon dioxide and 1 ATP molecule, 3 NADH and 1 FADH2 molecule are produced in each cycle. Okay, you know, we didn't mention oxygen here, so it doesn't need oxygen since all the electrons and protons are removed during oxidation of carbon atoms and received by NAD and FAD molecules. Okay, the electron transport chain, at the end of Krebs cycle, we ended up with a reduced NAD and FAD in the form of NADH2 and FADH2. These interact with coenzymes in the mitochondria on the inner side of the membrane of the mitochondria. This is called the cytochromes. These continue handling over the NADH and the high energy electrons are passed from one molecule to another, from one cytochrome to another. Then it's released to form ATP from ADP plus phosphate group, as you can see here. As it's transferred, ATP is formed. This process is called oxidative phosphorylation. So the NADH and the FADH2 resulted from the Krebs cycle will be moved down, see, from molecule of cytochromes until, and during each step producing ATP on the way, until it reaches actually the oxygen, which is the last receptor in the electron transport. Hydrogen and oxygen form water. Okay? In electron transport chain, each NADH molecule produces 3 molecules of ATP, while each FADH molecule or FADH2 molecule, produces 2 molecules of ATP. Okay, so if you calculated 2 molecules of ATP produced on the outside and 36 molecules of ATP on the inside in the mitochondria, so that's a total of 38 ATP formed. Okay? Now, if we talk about anaerobic respiration, it means respiration without the presence of oxygen. This is done by use of enzymes and is called fermentation. It's done in the presence of special enzymes. The anaerobic respiration starts the same way as the aerobic respiration, by decomposition of glucose molecules into 2 molecules of pyruvic acid with 2 molecules of NADH and small quantity of energy as 2 ATP released. Okay, pyruvic acid then converted according to the cell type in which it was formed. In case of animal cells, the muscle fibers, where muscles exert vigorous efforts or exercises and consume a lot of oxygen, most of the oxygen in their cells, and tend to convert to pyruvic acid, which is anaerobic respiration. It converts pyruvic into lactic acid and combining with hydrogen on the NADH. So what happens during muscle exercise? Accumulation of lactic acid is forming from converting pyruvic acid to lactic acid. This is known as muscular fatigue. If oxygen is available, lactic acid is converted into pyruvic again. Understand? So the lactic acid is a form of storage of pyruvic acid. This is what makes the muscle cramps, by the way. This is the acid responsible for the muscle cramp. In the case of bacteria, pyruvic acid is converted into lactic acid. In case of yeast or some plant cells, pyruvic acid is reduced into ethyl alcohol and carbon dioxide, as you can see here. This is another type of fermentation called alcoholic fermentation, and is used in the industry of some products. Well, I hope you got the maximum benefit from this lecture, and thank you very much for watching.